This lecture covers Chapter 1, Picturing Distributions with Graphs, for the Introductory Statistics course, Math281. In Picturing Distributions with Graphs, we're going to go over a set of objectives. We'll go over definitions of a lot of different terminologies, including individuals and variables, the two different types of data, categorical and quantitative, and learn some other names for them. We'll look at ways to chart categorical data using bar graphs and pie charts. And we'll also look at ways to chart quantitative data, which would include histograms, dot plots, and stem plots. We'll also look at how do we interpret histograms, and then also looking at graphing time series using time plots. We're not going to focus on exactly how to create these graphs in this particular lecture. We're going to look at graphs and learn how to create them by hand, and then we'll have another lecture that describes the technology behind creating those lectures, or creating those graphs. So let's go ahead and get started looking at individuals and variables. The first thing we'll start with is looking at individuals. Individuals are basically the subjects that are part of an experiment. In this particular example, we're talking about individuals as objects described in a data set. And again, another name for an individual would be a subject or an object in a particular data set. A subject would be if somebody called you and asked for your opinion about the upcoming presidential election. An object would be perhaps if we observed traffic in a particular intersection and we looked at whether a car ran a stop sign or didn't run a stop sign. So again, individuals are the objects described in a data set. Individuals could be people, animals, plants, things, anything that we're studying. Some examples of individuals would be freshmen, newborns, golden retrievers if we were looking, let's say, at hip dysplasia, fields of corn, looking to see if they had smut on them or not, particular cells. Now would be a good time to pause and take a couple seconds to see if you can describe some subjects that would be of interest to you in your area of study or even in any particular areas of interest that you have. A variable is any property that characterizes an individual, and basically a variable is what we are studying about a particular individual. A variable can take different values for different individuals. That's why it's called a variable, because it could vary. So for instance, for freshmen, I could be looking at age, gender, blood pressure, or blood type. For fields of corn, I could be looking at leaf lengths. I could be looking at a particular flower color as well, based on, let's say, another individual that I could put up here, like flower pots. And I could look at flower pots, and within those flower pots I could look at flower colors. So again, it's a good spot to pause again and write down some examples of some variables that you would be interested in looking at if you were to design an experiment in any area of your field of study or just an area of interest. Now when we talk about variables, we need to remember that there's two types of variables. So again, we're talking about variables. So the two types of variables that we are looking at, we will start with our first one to be quantitative. So our first type is quantitative. Now another name for quantitative is numerical. So you'll see these used interchangeably. Basically, a quantitative and a numerical variable are used interchangeably. Well, basically for a quantitative variable, it's some quantity assessed or measured for each individual. Then we can report the average of all individuals, for instance. So looking at that definition one more time, quantitative or numerical variable. It's some quantity assessed or measured for each individual in an experiment. We can then report the average of all individuals. So some examples here would be something like age. And this would have to be age in years. We wouldn't say your age is too old. That would not be quantitative. So age in years would be an example. Blood pressure would be an example. But again, it's not just blood pressure as being too high, too low, or just right, but it would be the actual numbers in numerical values listed. We could also look at the length of an object, let's say in centimeters. And these would be all examples of quantitative or numerical variables. Now, these would not be, for instance, your zip code. Now if you think about your zip code, Rapid City 57701, Spearfish 57783, and of course the ever popular 90210, we can record those as numbers, but it wouldn't make sense to report the average zip code. It might be making sense to report the typical zip code, but not the average zip code. So even though zip code is a number, it doesn't make sense to find its average, so it's not counted as a quantitative or numerical variable. So zip code, area code would be another one. It doesn't make sense to find the average area code. Uniform number. It doesn't make sense to find the average uniform number, even though it's a number as well. And another example would be your Social Security number. It doesn't make sense to find the average Social Security number. So even though they're numbers, it does not make sense to find the average. The other type of variable that we're going to look at is called a categorical random variable. So we'll jump down here. So the second type of variable that we want to look at is categorical. Now another name for categorical is qualitative, meaning it's basically a quality that I interpret. It's some characteristic that describes each individual. Now, once we find this characteristic for each individual, instead of reporting the average, which wouldn't make sense if we were talking about gender, blood type, or let's say whether there was a stop or not a stop at a particular stop sign, we can then report the count or the proportion of individuals with that characteristic. Now, it's important to be able to identify whether a variable is categorical or quantitative, because that kind of gives us an indication of what types of assessments we can do. But for now, let's go ahead and look at some examples of categorical or qualitative random variables. We could look at gender. So for gender, we'd be talking about male or female. We could be talking about blood type. So for blood type, A, B, and O. We could also talk about something like whether we tried to log in to your computer and either worked or didn't work. So log in, results, success or failure. And again, this list goes on and on and on. Now, remember up here where we talked about these ones that didn't work for quantitative, down here, zip code would be a categorical or qualitative random variable. So would area code. Okay, and maybe the first three digits of your Social Security number, like if it's 503 or 504 or 517, kind of indicates either where you were born or at least where you applied for your Social Security number. So these would fit into this particular category. Now, one very important skill in statistics is trying to decide whether a variable is categorical or quantitative. So how do you decide if a variable is quantitative and remember, quantitative and numerical are the same, or categorical? And remember, another name for categorical is qualitative. Well, first of all, you need to ask yourself a few questions. The first one to look at is, what are the n individuals being examined? And then, is it being examined from a sample or a population? So there's actually quite a few things going on here. First of all, this letter n represents our sample size, meaning how many people did we ask. For instance, if we were interested in determining whether students were wanting to have the whole week of Thanksgiving off instead of just the Wednesday, Thursday, Friday, instead of asking every single student on campus, maybe we could ask 30 students on campus. So that would be our sample size. Now, a sample is a subset of the population. So instead of asking every single student on campus, if we just asked a subset of students on the campus, that would be our sample. And of course, our population would be the individuals or the subjects that you are interested in. And these have some more definitions as well, and we'll talk about those more in depth, but that kind of gives you a good idea. So again, population are the individuals that you are interested in studying. And just as a quick side note, if you're using a population, I would use cap N to represent the population size. Lowercase n is for sample size, cap N is for population size. So that would be one of the first questions you would ask. Now, what is being recorded about those n individuals? And again, n here is sample size. Next, the thing that I'm recording, is it a number? And if it is a number, it usually is quantitative, but not all the time. Or is it a statement? You know, are we talking about blood type? Are we talking about, you know, whether a person has a cell phone or not a cell phone? But this is a case where we need to be very careful. Remember, there are some exceptions. Just because it's a number doesn't mean it's always quantitative. We have some exceptions here for this case, such as zip code, area code, social security number, etc. But again, those are exceptions. Those are actually categorical random variables, even though they're made up of numbers. So that's the first thing that we would do, is we would ask those particular types of questions. So let's go ahead and look at a data set, and with this data set, let's answer whether we are looking at categorical or numerical random variables. Okay, so here's our data set. First of all, I would look at here and I would say there's 1, 2, 3, 4, 5, 6, 7 samples, in this case patients. So n in this case would be 7. Now next I have diagnosis. Now the particular diagnoses that I'm looking at are heart disease, stroke, lung cancer, accidents, and diabetes. These are not numbers. Okay, they are basically a category of death, and so diagnosis would be a categorical random variable. Now remember, another name for categorical is qualitative. And we use RV to represent random variable, and we'll talk about the idea of randomness as well. The next variable we have here is age at death. Well, your age at death is certainly a number, and it's a number that it makes sense to manipulate arithmetically. It's find the average of. So this would be a numerical or a quantitative random variable. And again, RV stands for random variable. So for this case, this is our qualitative and this is our quantitative random variable. So next, let's go ahead and look at another study to see if we can identify whether I have qualitative or quantitative random variable, and then talk about the sample size, and then also talk about some other variables as well. Okay, in this example we're looking at a study that examines the condition of deer after a particularly tough winter. Basically in this study they're looking for the gender of the deer and the condition of the deer, and they're only looking at the conditions in terms of good and poor. They said they're looking at a random sample of 61 deer, and they show the raw data below. So this particular table gives us the raw data. And it's actually, as you can tell, not made up of 61 deer. These dots at the end meaning these observations continue on in the same type of a pattern. Now this over here is called a frequency table. And what we do with a frequency table is you take the raw data and you start tabulating. So for instance, individual 1 was a male and was in good condition, and so we'd make a tick mark here. Individual 2 was a female in poor condition, so that tick mark would go over here. Male poor, male good, male poor, female good, etc. And we would go on and continue this pattern until we had looked at all 61 deer, and I'd have my total of my tally marks over here, and in this particular case I'd have 10, etc. Now when I add these up, I have 10, 20, 30, 40, 50, 61. This should always total the total amount here. And in this particular case, that's n, lowercase n equals 61, because it is a sample. If it's a population, it'd be huge because the population of deer is quite large. So again, from the raw data, we can produce the frequency table. Now our first question here are basically the individuals. Who are the individuals and what are the variables, and are these variables quantitative or qualitative? So let's go ahead and talk about the individuals. What is being studied? Well, in this particular example, the individuals are the 61 deer. And then for the variables, I note a couple of things. I note the gender or sex, and then I also note the condition. Now both of these are things that are observations. They're not numerical, and so what I can talk about here is the type of variable. Well, these are both categorical, and another name for categorical is qualitative. So same for condition is categorical or qualitative. Now when I have an example like this where I have categorical or qualitative random variables, I can come up with a frequency table, and then I could find percentages if I wanted. So for instance, 10 over 61, 10 over 61, 23 over 61, and 18 over 61 would make up the percentages and the relative frequencies. So let's produce that table as well. Okay, to get started, this is a count or a frequency. So another name for count is frequency. Then I could look at relative frequency and then percent frequency. So if I looked at 10 over 61, I would get about 16%. And of course 10 over 61 would be about 16% here. 23 over 61 is about 38%. I'm rounding these. And 18 over 61 would be about 30% as well. Now when these add up, I should get 100%, so let's check it out. 16 plus 16 is 32, plus 38 plus 30% is 100%. And so another name for this table is not only a frequency table, but it also is giving us the percentage or the relative frequency as well. And then from here I can go ahead and produce some graphs depending on what I'm interested in. So when we're looking at categorical data, there's some graphs that apply particularly well for representing categorical data. The first type that we're going to look at is a bar graph. Now for a bar graph, each characteristic, in this particular case gender or condition or level, is represented by a bar. The height of the bar represents either the count, the tally, kind of like we looked up here in this particular table, or the percent, also as we produced in this table, with that characteristic or the relative frequency. So a bar chart for the data that we just looked at could be one of two types of bar charts. It could give gender, male, female, or it could talk about condition, and the two different types of condition that we could look at were good or poor. Another type of graph that does a really good job representing categorical or qualitative data is a pie chart. And these are pretty familiar. You see pie charts a lot in newspapers and magazines to represent the outcomes of a survey or another type of sample. Now a pie chart you have to be a little bit careful with. It can only represent how one categorical variable breaks down into its components. So for instance, the condition of the deer is good or poor. I can only work with that. I can't work with the male, female, gender, and the good, poor. So when I talk about a pie chart, it only represents how one categorical variable breaks down into its components. And basically we find the percentage that each component takes up, and then we take that percentage times 360 degrees, and that is represented by the slice, and the size of the slice again is tied back to the percentage of that particular category. So let's go ahead and look at some examples of these types of graphs. For this first example, we're asking subjects whether they like carrots, peas, or spinach. And so each response would be a yes or a no. So this is an example of our raw data. And then we take the raw data and we tally our values, and then we try and produce some sort of a numerical representation graphic for that, like a pie chart or a bar chart. But we have to figure out which one will work where here. Now for this particular case, if you notice, this first subject, he likes all three of these. Subject 2 only likes carrots, and so when I try and find the percentages under carrots, I say I have 1, 2, 3, 4, 5, 6. So I have 5 out of 6 of the people like carrots, and then the other 5, 6, sorry, the other, yes, 2, 6 or 1/3 don't like carrots. So 4, 6 and 2, 6, and there's my percentages. Now notice this does sum to 100% here. Now same with peas, it sums to 100%, and the same with spinach sums to 100%. Now we can only produce a bar graph of this though. I cannot do a pie chart. A pie chart wouldn't make sense here because when I talk about the percent who like, if I add these up, I get more than 100%. So if I took 67 and 50 and 33%, I would get more than 100%. So I can't really do a pie chart here to represent all of this data. If I wanted to, I could do individual pie charts for carrots and do pie slices for just like and don't like carrots, another pie chart for peas, and another for spinach. But I cannot produce a pie chart that represents all of this data at once. Let's go ahead and look at another example as well. Now in this case we're asking a similar question. So again, this is another example. We're asking a similar question. We're not asking about each individual vegetable though. We're just saying which one do you prefer, and the person selecting can only choose between peas, carrots, or spinach. They can't say yes or no to each one of those variables. So in this particular case I have half of my 6 subjects, 3 out of 6 subjects that prefer carrots. Okay, 33% prefer peas and 17% prefer spinach. And if you notice, these sum to 100%. Now here I have 2 cases that I can go with for a graphical display. I could go with a bar chart, which is what we have here, and or I could go with a pie chart. Now again, the second table shows the answers to just one question. So that is the reason we can display the results in either a bar graph or in a pie chart. Now if you notice, if I look at these percentages, they do sum to 100%, and because they sum to 100%, it makes sense to also create a pie chart. So the difference here is in this first case, the first table shows the answers to 3 separate questions. Do you like carrots? Do you like peas? And do you like spinach? We can use one bar graph to display all the results at once. We couldn't use a pie chart because there's more than one categorical variable here. So again, be careful with what you use to produce a pie chart because sometimes it doesn't make sense. Your data might not match the structure that's needed for a pie chart. Okay, let's go ahead and look at one last example to see how we can analyze a graph. And when we analyze a graph, we're looking for some information. We're looking for the individuals. We're looking for whether it's categorical or quantitative data, and we're also looking for whether I can use a bar chart or a pie chart, which would make more sense. Okay, so here's our graph. Let's see what we can come up with. First of all, the heading on this graph is we're looking at the percent of current marijuana users in each of 4 age groups, and this is from data in 2004. The vertical axis is giving us the percentage who are current users, and the horizontal axis are giving our age groups. And so first of all, who or what are the individuals? That's the first question we want to ask. Well, when we look at this, I can't really tell about a total or a count. I can add up the percentages, but that doesn't really make sense in this case, and I have no idea of the number of people that are asked. So basically the individuals look to be Americans because that's the heading here. Basically the Americans that are 12 or older. And the reason I'm saying that is on our horizontal axis here I go 12 to 35 plus, so 12 or older. Next thing we can do are talk about the variables. So variables are basically age group. So you'd ask a person, which age group do you fall in? 12 to 17, 18 to 25, 26 to 34, and 35 plus. And then we'd also ask them whether you use marijuana or not. So marijuana use. Now both of these in this particular instance are categorical. Now remember, another name for categorical is qualitative. Well, clearly this is a bar graph. We could not represent these data into a pie chart because we've asked about 2 different basic variables here. We've asked about your age group, and we've also looked at whether or not you use marijuana. So a pie chart could only display the results for each group separately. I couldn't combine these. And then also, if I ballpark these, I'd have about 8% here, 17% here, 9%, and let's say 3%. And those don't come to summing to 100%. So I can clearly see that a pie chart would not work here. Next, let's go ahead and move on to common ways to chart quantitative data. Now when we talk about common ways to chart quantitative data, it might be a good idea to kind of review what quantitative data means. Quantitative and numerical are interchangeable, and remember it's a numerical response that makes sense to find the arithmetic mean of. So some common ways to chart quantitative data. First of all, histograms are very common, and this is a summary graph for a single variable. Okay, I'm only asking about one variable. And histograms are useful to understand the pattern of variability in the data, especially when you have these huge data sets that you're trying to make sense of. Now, a dot plot and stem plot are graphs that basically contain all the raw data points. All the original values of the data set are represented here. And they're really nice for small data sets, and they're really easy to do by hand. And again, they look for patterns and variability in the data. Now up here, let's jump back to histograms a little bit. When I talk about a histogram, a lot of times I lose individual data points because I categorize them together. So in the previous screen we looked at age groups. Well, in these age groups, I don't know, do I have a 13-year-old that uses marijuana here? I don't know. All I know is I have about 8% of 12 to 17-year-olds use marijuana. So again, I lose my individual data with the histogram. Dot plots and stem plots maintain that individual data. I can still see the individual data values. Line graphs and time plots are very commonly used as well. And the main part about a line graph or a time plot is keeping track of how things changed over time. Meaning, if I start collecting data in 1980, I want to compare what happens from 1980 to 1981. So my horizontal axis will keep that time in order because, again, that makes sense. The first one that we're going to look at is producing a stem plot. So there's a number of steps that you go through when producing a stem plot. And again, it's going to be important that you read through the examples in your book because there's a lot of different types of stem plots that we can look at. When you start producing a stem plot, the first thing you want to do, and I'm going to be working with this data set down here, is I want to separate each observation into a stem consisting of all but the final rightmost digit and a leaf. And the leaf is that basic remaining final digit. Stems may have as many digits as needed, but each leaf contains only a single digit. So for instance, when we look at the number 9 down here, I would separate that into 0 and 9. And again, 9 would be separated into 0 and 9. 22 would be 2 for my stem, 2 for my leaf. 32 would be 3 for my stem, 2 for my leaf. 33 would be 3 for my stem, 3 for my leaf, etc. And you can see the pattern here. Next thing I do then is I write the stems in a vertical column with the smallest value at the top and draw a vertical line at the right of this column. And that's what I've kind of already done here. But what we want to do is we want to look at the smallest value, and it's 9. So the stem on that was 0. And then the largest stem looks like it's a 4. Wait, here we are clear at 7. So I'm going to go 0, 1, all the way up to 7. Now I can't skip the stems that are not used. For instance, 1 is not used. I can't not include it because I need to have a sense of where the gaps are in the data. So these are the stems. And then I draw that vertical bar to separate the stems from the leaves. And then what I do is I write each leaf in the row to the right of its stem in increasing order out from the stem, and they have to be spaced evenly as well. So the only values I have here that go in the 0 stem are 9. So I have 0, 9, and 0, 9. And each of these represents the number 9. I don't have anything in the 1s column. And then in the 2 column I have a 22. And then in the 3 column I have quite a few. I have a 2, 3, 9, and a 9. And again, I want to keep these evenly spaced. In the 4 column I have a 2 and a 9, 42 and 49. And then I have 52 and 58. And then I don't have anything for the 6s, and then I skip up to 70. And I cross these out as I go because I want to make sure they're represented. Now let's look across here in the 3s column, or the 3s row. I want these to be in order from smallest to largest because it gives me an idea of the data. So I can do a lot of different things with the stem and leaf plot, but whatever I do, I need to remember to include a legend that explains what's going on here. So for instance, 3/2 equals 32. You might say, well, duh, what else could it be? But 3/2 could be 0.32. I'm not sure, and so again, that's why we always have to represent this. Now, variables with a wide range might be rounded or truncated. So for instance, if I had something with 8 decimal points, I might want to round that so I can look at a few more manageable stem plots. And I can also split these stems to show greater detail, and your book gives you some really good examples of that. Next, let's go ahead and look at some different types of stem plots and how to analyze them. So in this stem plot example, we're going to look at how fast do skin wounds heal. So for this example, biologists measured the rate at which new cells closed a razor cut made in the skin of an anesthetized newt. Here are the data from 18 newts measured in micrometers per hour. So this particular newt had 2/8 or 2/8 micrometers per hour. This one was 1/2, etc. Now before we get started creating a stem plot here, the first step I need to do is to sort the data. That helps me keep track because in the stem plot the data has to be in order. So let's first of all sort the data. It's usually best to sort the data in order from smallest to largest because that's how it will appear in the stem plot, or another name for stem plot is a stem and leaf plot. The second thing that we'll want to do is assign the values to stems and leaves. Now after we assign the values to stems and leaves, they're already going to be in order, and I can produce my stem and leaf plot. So if we notice here, my stem values are the tens position. Looks like they go 1 through 4. So here's my stems. Now over here will be my leaves. So in this case, 1, 2, 4, 8, of course evenly spaced, will be the leaves across from the one stem. And then working across here for my 2s will go 2, 2, 3, 3, 6, 7, 8, 9. And again, these have to be evenly spaced. Across from the 3s would be 0, 3, 4, 5, 5. And again, as we're keeping track, it's a good idea to cross these out to make sure 3, 4, 5, 5 to make sure they're all represented. And then our last one is 40. Now, of course, the legend is necessary here because we need to know, are these values that are 1/2, is that 0.12 or is it 12? And in this case that equals 12. So a legend helps us see that. Now pretty soon I'll start to analyze the data, but for the most part it looks like most of my data lies in the 20s, or in the 20 micrometers per hour. So it kind of gives us a good view of the data. My values range from 11 as a minimum and 40 for a maximum, so that gives me an idea of the range or the variability. The next technique that we're going to look at, or the next type of graph that we're going to look at, is creating a dot plot. Now remember, stem and leaf plots and dot plots are usually best for smaller sample size. Now when we go through and we make a dot plot, the first thing we want to do is to create a single axis representing the quantitative variables range, and this basically means a number line. Then we represent each data point as a dot positioned according to its numerical value. When two or more data points have the same value, then we stack one on top of the other to represent the frequency. So let's go ahead and look at the same data that we just did, and we'll look at the raw data, then we'll look at the sorted data, and then we'll produce a dot plot. So the first thing that we did is we took our raw data, and then this was the wound healing, don't forget, and we sorted it. Sorting the data just helps you keep track of things. Now what I want to do is produce my number line. So again, this is just a number line that keeps track of all the values. Now there's no reason to have 50, 60, 70 on this number line because my largest value only goes up to 40. So the first thing I do is I note number 11, and I note that that goes in between 10 and 15, and I try and kind of scale that the best that I can. Here's 12, here's 14, here's 18, and then 22 has two values, or two instances of 22, so I stack one on top of the other. And then this produces my dot plot. And this dot plot gives me a pretty good idea of the spread of the data, the frequency of the data, and kind of where most of the data is clustered. And again, stem and leaf plots and dot plots are better for small data sets. Next we're going to look at making a histogram. There's quite a bit more detail involved in making a histogram and more choices that need to be made. And again, reading through your text will kind of give you an overview of some different types of histograms that we can look at, and then also some different types of stem and leaf and dot plots as well. So to get started with making a histogram, the first thing you do is you need to look at your raw data. Now for the raw data, the range of values that the quantitative, and again a histogram is based on a quantitative random variable, the range of values that the quantitative variable takes is divided into equal sized class intervals. So for instance, when we looked at the age range of marijuana users, for a histogram we'd want the ages to go 10 to 15, 16 to 20, etc., and splits up evenly by that. Now these are called classes, and these make up the horizontal axis. The vertical axis is represented either by the frequency or the count, remember when we made the tally marks for the deer population, or the relative frequency, which is the percentage. Now these do not change the shape of the graph whether I use the frequency or the relative frequency. Then for each class, basically you draw a vertical column, and the height of the column represents either the count or the percent of data points that fall into that class interval. So let's go ahead and look at a raw set of data, and based on that raw set of data, let's go ahead and produce a histogram. So for our histogram example, we're going to look at guinea pig survival time in days after inoculation with a particular pathogen. So first of all, we're looking at a sample size of 72 guinea pigs, and then for each guinea pig we look at how long they survive from the time that they were inoculated with this pathogen until they die. So let's start here by looking at the raw data. Now this raw data is nicely ordered, so that kind of gives us a little bit of an idea of what's going on here. It looks like the minimum survival time after inoculation was 43 days, and the maximum survival time after inoculation was 598 days. Now let's go ahead and build a histogram with class size of, let's say, 50. We could have done a class size of 10, and we could have gone 40 to 50, 50 to 60, etc. But we'd have so many classes, so I want to start with 50. It might not be the best choice, but let's go ahead and start with a class size at 50, and let's go ahead and start at 0. So 0 is included in the first class. So we can use some software like Excel or Minitab to produce these graphs, and again I'm not focusing on how we produce these graphs, we'll do that in a different lecture, but for now I just want to look at how we use and interpret these graphs. So to get started, first of all, I take the raw data points and I have my first class width, which is from 0 to 50, 50 to 100, etc. And in this particular case, 0 is included in the first class. So if I have an observation that's exactly 50 to 100, I will include it in this class. So where would you include an observation that was exactly at 100? You'd include it to the right. So how many observations do I have between 0 and 50? I have 2, and so the height of my bar here would be 2. How many do I have between 50 and 100? Looks like I have quite a few here. Between 50 and 100 would be the rest of this first row, and then the second row, and then the question is, do I include that 100? And the answer is no, you do not include 100 in this category. That 100 goes to the right, so it goes 100 to 150. So I have 28 here, and those values of 28 would be these values right here. They're all between 50, including 50, and 100, but not including 100. And then we'd split them up and we'd look at each category like this. Now, this particular distribution is going to be called a right skew distribution and unimodal. So those are some words that we can use to describe this. So again, unimodal. Una, of course, means one. Modal means a peak. So unimodal means I have one large peak, and this is a right skewed or a positive skew graph. That means the bulk of the data is to the left, the tails extend out to the right. So again, right or positive skew. And we'll talk a little bit more about this modality, unimodal, bimodal, and skewness as well. But to get started, this is how we'd produce our graphs. Now if I wanted to, I could have done a histogram that went 0 to 100 and combined all these values. And there's no right or wrong way for number of classes. It's usually just you look at the different types of graphs that you get from the classes, and again, software does this for you, and it makes it really easy to determine, what is showing the data and what is not. Now another interesting thing to note is on this axis, because I'm just looking at counts, this is my frequency. It's just a count. If I were to go through and tabulate these values and say, okay, how many fall within 0 to 52 out of the total of 72? So 2 divided by 72 would give me a frequency, or sorry, a relative frequency or percentage. This height would be the same percentage. So the frequency graph and the relative frequency or percentage graph are identical. And I'll give you a quick picture of the relative frequency graph that goes with this. So this shows us the frequency distribution, and then down here is the relative frequency or percent. And because I have the same classes and it's based on the same data, it has that same general shape. The only thing that changes here is, again, the vertical axis. This is percentages or relative frequencies, and these are their frequencies. Now there are two histograms of the same data set here using the same choice of classes. The histogram on the top displays the relative frequency, or sorry, the one on the bottom displays the relative frequency or the percentage of guinea pigs that survived a particular amount of time, and the histogram on the top displays the frequency or the actual count of the guinea pigs. And again, notice the shape is unimodal and right skewed for both of these graphs representing that data. Now one of the important things we need to talk about here is how do you choose the classes for a histogram? How do you choose the number of classes? And basically it's an iterative process, try and try again, and we have software that does this for us, so it kind of makes it a lot easier than having to draw them out by hand. Basically you do not want too many classes with either 0 counts or 1 counts in them. And you don't want to overly summarize so that you lose all of the information, meaning have just like 2 spikes on your graph of counts of 50 in each one. Not so detailed that is no longer a good summary. So a couple rules of thumb: try starting with 5 to 10 classes, then refine your choice. And remember, there isn't a unique or perfect solution to this, so you're not getting it wrong, but you could be losing some information from your data based on the number of classes that you choose. Well, now that we know how to build histograms, let's go ahead and move on to interpreting histograms. Now we've already talked about interpreting histograms just a little bit in our last graph that we looked at, but there's basically some information that we talk about for every type of histogram. The first thing we do is we look for the overall pattern and for striking deviations from that pattern. So look for the overall pattern, and then also look for whether or not there's any striking deviations. So sometimes there's a slight deviation, but really we're interested in the ones that are extreme deviations. And then we talk about the shape, and we talk about its center, and then we talk about spread, and then these striking deviations would be possible outliers. So those are usually the things that we look for. So the shapes that we can talk about, they could be unimodal, meaning one mode or one peak. They could be bimodal, meaning two peaks. And of course trimodal would be next on our list, but usually if it has three or more peaks we go with multimodal. And then we could also talk about, with its shape, whether it's symmetric, right skewed, kind of running out of room here, right skewed, or left skewed. And we'll talk about these in a little bit more detail. Center, we're looking for the approximate center of mass. Where is the middle? Now there's two different types of center that we could talk about. One is the mean and the median, but for this graph up here we can see that, you know, it looks like the bulk of the data is over here, but if we were to balance this like a teeter-totter, these values out here would be having a lot of influence as well. So I'm guessing the center of mass is somewhere in this bulk, but maybe pulled a little bit this way because of that teeter-totter approach. Now when we talk about the spread, we are talking about the range of values taken on by the variable. So the range of values taken on by the variable. So for here it looks like we go from about 0 to 600, and we know that it actually starts in the 40s, but this histogram doesn't show that, because a histogram you lose the original data. Now for outliers, basically outliers are individual values that do not fit the overall pattern. So in this example that we have right up here, the bulk of the data is here. This doesn't seem too extreme, but these points right out here do seem, you know, like they do not fit the overall pattern at all. So first of all, let's go ahead and talk a little bit more about shape. Now, the shapes that I'm going to be looking at right now are unimodal, and these are some of the most common unimodal shapes that we're talking about. The first one is symmetric. Now, a very common symmetric distribution that we're all probably fairly familiar with is the bell-shaped distribution. Symmetric basically means if I fold it down its center, it's the same on the right as on the left. Not every symmetric distribution has to be bell-shaped. For instance, I could have a uniform distribution that's symmetric. I could also have a bimodal distribution that's symmetric, but these are just some examples. Let's also talk about a left skew distribution. Another name for left skew is negative skew. Basically, the left side extends much further out than the right side. So the left side has these extreme values that extends further out. So when I think about left skew, I think about where the outliers are, or potential outliers are. So the skew is always in the direction of the tail, where the extreme values are. So we've already talked about right skew a little bit. Now another name for right skew is positive skew. And again, this is just the counter to the left skew. The right side toward the more positive numbers, the right side extends much farther out than the left side. And it's going to be important that you be able to talk about these different shapes of the distributions and also draw graphs that are right skew, left skew, etc. So again, potential outliers are on the right. Values that are not toward the bulk of the data. So all of these are unimodal and they're all single peaked. And again, the side of the skew is the side of the tail of the distribution. The next thing that we're going to do is to look at some general shapes of histograms and then start to describe the shape of these histograms. Okay, so let's go ahead and look at this first example. Now, first of all, this should look familiar to you from reading in your text. This is the young women with anorexia. So this is the age and years at the onset of anorexia, and this is the frequency. Now, if we had to talk about this particular graph, this seems to be kind of a middle value with 3 bars on either side. Now, these 2 bars look fairly symmetric. These do as well, but now these don't. But in general, the overall shape here, I think if we were to describe this, is roughly or fairly symmetric. So I'm going to say roughly symmetric. And I also kind of think that it's unimodal. And you might say, well, wait a minute, there's all these modes. Well, I think unimodal kind of represents this peak value. So I think this is a good example of a graph that's roughly symmetric and unimodal. Let's go ahead and look at another example. Now for this graph, I kind of cut off the horizontal axis here, but this is the servings of fruit per day. And this again should be familiar from your reading as well. So 15 people, number of subjects, 15 people have 0 servings of fruit per day. And it looks like 11 have 1 serving of fruit, etc. Well, if you talk about the bulk of this data, the skew looks to be in the right direction. And so the bulk of the data is on the left. And so I think that this one is right skewed. And I also think another way we could describe this is unimodal, because the bulk of the data is here. Now if you wanted to get picky about it, it's actually probably bimodal because of these 2 peaks, but I kind of look at this and think I have one large distinct peak right there. So again, not all distributions have a simple shape that I can always say is exactly symmetric, right skewed, left skewed, unimodal, or bimodal, but a lot of times I can get a general idea of the shape of the graph. Let's go ahead and look at one more example. So for this example, we're looking at, again, this is another graph from a data set in your textbook. This is the fish and perch example. We're looking at the fish that have a length that looks like 5 to 10 here would be a count of 1, 10 to 15 centimeters would be another count of 1. And if I'm talking about this particular graph, it's not as obvious. It does kind of look to have 2 distinct modes, so it looks like there's a mode here and a mode here, and they're not the same height, but they're definitely 2 distinct peaks. So I would call this a bimodal graph. I wouldn't say it's symmetric, and I wouldn't say right skewed or left skewed. I would just call this complex. So this would be a complex bimodal graph. It doesn't have any particular type of skewness or symmetry. The next thing we're going to talk about is outliers. Now, an outlier is something that deviates a lot from a pattern that we see in the data. So an outlier is an important kind of a deviation. It's an extreme value. Outliers are observations that lie outside the overall pattern of the distribution, and they could live outside to the right or outside to the left. They could be extremely small or they could be extremely large. And when you're analyzing data, you should always look for the outliers and try and explain them. You don't just ignore them. There has to be an explanation for them. So let's go ahead and look at a graph that has some outliers. Whoa, it's an outlier because it's such a huge graph to start with. Let's see if we can get it shrunk down here. And in this particular graph, we're looking at basically elderly in the population, so the percentage of state residents age 65 and older. So the bulk of the data looks to be here, and if we were to just look at this part of the data, I would say it looks symmetric and unimodal. if we just talked about this centerpiece. Now Florida looks like between 18 and 19% of their population. I think Florida is the only one here. One. Yep, just the height of one here. So 18 to 19% of their population are age 65 or older. So this looks to be extreme large, and Alaska looks to be extreme small. And again, this large gap that's between the bulk of the data and both of these states, Alaska and Florida, kind of show that these are outliers. So again, an outlier we don't really have a rule for right now. Pretty soon we'll come up with something called the 1.5 IQR rule, but for now you kind of just analyze the data and you kind of look to see something that deviates from those patterns. The last topic that we're going to look at in Chapter 1 is graphing time series. Now when you graph time series, the most important thing to remember is time goes on the horizontal axis and it goes in order that makes sense, meaning if we start in 1960 with something, we want to start with 1960 and then go to the next year. We don't want to scramble the years or anything like that. So let's go ahead and look at graphing time series. When we graph time series, data is collected over time and they're displayed in something called a time plot or a time series plot. And again, time is on the horizontal axis and the variable of interest is on the vertical axis. Now the variable of interest could be output or production, or it could be time, it could be temperature, and that's along the vertical axis. Then we look for a possible trend, or which is an overall pattern that's obvious. And then we also can look for cyclical variations. So for instance, our cold temperatures are often in January, or our warmest temperatures are often in August. And so those are some things that we can look at. Let's go ahead and look at a graph so we can analyze it just a little. Okay, pop the graph in. Let's go ahead and talk about some characteristics of this graph. This is the monthly atmospheric CO2 levels recorded at the Mauna Loa, Hawaii Observatory between March 1958 and August 2009. So when we look through this, it looks like every March they have marked on our horizontal axis and then the years increase, which would make sense. And another thing that we can see here is we notice a clear upward trend. This is indicating a gradual increase in monthly CO2 levels, but we'll notice within the overall trend of increasing, I do see these decreasing spots too. That means I have cyclical variations. We observe lots of month-to-month variability, but these variations look to be quite regular and predictable. So decrease, increase, decrease, increase, and this pattern continues with an overall increase in this particular graph. There are as many peaks as there are years in the data set, so there's seasonal variation as well. The yearly peak CO2 level is in the late winter or early spring, it looks like, because here's March to March. Okay, and then we can also look at this and we can talk about the overall trend here looks to be linear, meaning I could possibly sketch in a straight line that predicts this, but maybe a curve would be a better model for prediction, and that's another thing that we'll talk about a little bit later on. Let's go ahead and look at a few other graphs so we can talk about their time series plot. So for this graph, it looks like we're talking about the salinity in the Shark River water in the Everglades National Park over a 7-day period. So the 7-day period is in 2009, and the first day is September 30th, October 1st, etc. So we can follow this here. Now this time plot doesn't look to show an overall increase or decrease. It looks like it's remaining fairly stable over time, but I also do see these cyclical patterns. So first of all, when we're talking about water salinity, that's a quantitative variable. Now note that the data collection is done, it looks like to me, fairly often here. So it looks like in one day, it looks like they do a data collection, 2 or 3 times. We see no evidence again of an overall trend over such a short period, but we do see a cyclical pattern that's pretty obvious. Looks like salinity peaks twice a day. It peaks here and here, twice per day. Now, corresponding with the times of the tide in the Gulf of Mexico ocean water, that would kind of make sense as the tides come in and out. And again, here is something we don't know from just glancing at the graph, but if you read your textbook, the data were collected near the mouth of the river, and so when tides come in, the salinity from the ocean would come in, and it would make sense that that would peak. Okay, this graph, if you read the figure 4 at the top, these are cesarean delivery rates by gestational age in the United States from 1996 to 2000. Now this top line means a C-section was performed at less than 34 weeks, 34 to 36 weeks into the pregnancy, and 37 weeks or over. The vertical axis is per 100 births, and then down here on the horizontal scale I do have time. So a couple things that we note here. I'm keeping track of 3 different subsets. Now here we have 1997. I'm keeping track of the number of cesareans per 100 in 1997 that were performed less than 34 weeks, 34 to 36, and 37 weeks in the pregnancy. So this graph is actually providing quite a bit of information. So again, time plot of C-section delivery rates in the United States. The data are annual rates. There's a clear upward trend for each one of these. So again, it looks like to me that each one of these is increasing. Now it's a fairly gradual increase in the proportion of births delivered using C-sections. And what we're looking at here is a truly visible trend in all 3 groups. I don't see any cyclical variations, but there definitely is a trend. To finish up time series plots, we need to talk about the scales of these graphs. So basically what we need to remember is that scale matters. How we scale our horizontal axis and our vertical axis definitely matters in how we interpret the graphs. So let's go ahead and look at a lot of graphs and then talk about the effect of scale on each of the graphs. So take a second to look through all of these graphs. How you stretch the axes and choose your scales can give a different type of an impression about what's going on with the data. Now all of these data are absolutely identical. Okay, so each of these graphs represents the same data set. So let's look at a few characteristics. First of all, these are death rates from cancer in the U.S. from 1945 to 1995. Now if you look at the horizontal scale here, each of these goes from 1940 to the year 2000. Now our data only is collected till 1995, and that's why it doesn't go quite to the end to 2000 there. So our horizontal scales are all identical here. Now our vertical scales are what's changing. In this particular case, our vertical scale goes from 0 to 250, as does this one. Okay, as does this one over here. This is the odd man out here. This scale starts at 120 deaths per thousand, death rate per thousand, and then jumps up to a little over 200. So this is the odd man out here. Now if you look at this graph, it looks like there's a slight increase, but a very, very gradual slight increase. You know, nothing to be concerned of type of thing. Now over here, same data, I'm looking at this graph and it looks like there's a drastic, extreme increase. So just by changing the scale, not starting at true 0, there's a huge change here. Now another thing to consider is all the blank space in this particular graph. So these two graphs are identical. Now this one is just stretched vertically, meaning I took the graph space and stretched it more vertically. And we can see that the trend here, very small, gradual increase, but over here again I see that extreme increase. Okay, now over here, this particular graph doesn't make a huge use of that space either, but it shows that nice increase as well. So this almost shows flat line. All of these show some sort of a stronger increase. This shows a very great increase just simply by changing the scale. And so the things I want you to think about here are, when you change the scale, are you changing it to show something in particular? Are you trying to exaggerate something to show an extreme increase? Or when you shrink your scale, are you trying to hide something? So these are things to keep in mind for Chapter 1. We looked at all sorts of graphs, and we'll look at producing these graphs using Minitab, Excel, and remember those manuals are on the student resources in your Stats Portal website. Hopefully you enjoyed learning about Chapter 1 and graphing data.